lymph. What is lymph composed of? What is it like, similar to? Real similar to plasma. So it doesn't have the big red blood cells from the blood. It doesn't have big things like albumin or the big chunky things because that stayed in the blood. Lymph is created at the tissues. So any of the fluid that escaped the capillary bed that's out in the tissue, lymph is this extra drainage system removing that from the tissues. If it has any tissue debris, there might be antigens in the area. Maybe it's a site of infection that's being drained. Maybe it's a post-surgical site. Maybe it's a swelling from something. So that's going to be what's drained out this fluid is. And so it goes through a series of lymph nodes to provide our immune system an opportunity to inspect in case there are antigens before we send it into the blood and let it loose throughout the whole entire body. So it's a way to try to compartmentalize any particular antigen from mobilizing too much to our body. So the composition of lymph is going to be really clear, watery. It's going to contain mostly water, some immune components, obviously from the lymph nodes, but it doesn't have the main large protein structures that's found within the blood. So we have lymph vessels that function really similarly to veins because they're going to be low pressure. They're bringing the fluid back towards the heart. They have valves to prevent backflow. The only difference being it's running through a series of lymph nodes. We have areas in our body where there's a higher concentrated number of lymph nodes in our inguinal region, in our axillary region, or our cervical region. So those are some clusters, but we have lymph nodes in lots of other places, not just those. Those just are clusters of them. Lymph is returned to the blood via the subclavian veins. The majority of lymph is returned via which duct into which subclavian vein? So the majority, 75% plus of our lymph comes back via the thoracic duct into the left subclavian vein. And then the remaining comes into the right subclavian vein. The lymph nodes, you should be able to spot one. Histology of a lymph node. There's a medulla in the middle and the cortex on the outside. They've got germinal centers. What type of cell would be in the medulla region, part of our specific immune system? T cells. And why is that? Yeah, so T cells require cell-to-cell contact in order to eliminate an antigen. So that wants to be on the medulla where most of the fluid is coming through. What type of cells would be clustered in those germinal centers out in the cortex? Plasma B cells first, because they make the antibodies, and the antibodies then can be sent down into the medulla region. Spleen is located where? The spleen is located in your left upper abdominal region above your kidney. It is just a round organ that blood filters through. What is the two jobs of the spleen? Removing the old red blood cells. What part of the spleen does that? Red pulp, yep. And then what is the other component of the spleen? White pulp, and that is inspecting the blood for any circulating or blood-borne antigens. That's the job of that. Then the thymus, where is that located? In the chest. So we're right in the thoracic cavity. What is the function of the thymus? To mature lymphocytes, so you have your just Joe Blow lymphocytes from the marrow into the thymus, and then they become T lymphocytes. So they are not T cells before they were in the thymus, but they are indeed after they were in the thymus. But they are naive T cells when they leave the thymus, meaning they're a T cell, but they still have yet to be coded for a specific antigen. You have to have some sort of exposure to an antigen in order for these sensitize into something that they're coded for. Who can tell me what MALT is? Yep, so mucosa-associated lymphoid tissue. Where is MALT located? Tonsils being one of the places. or respiratory tract, any place that's got mucosa, GI tract even. The GI tract is called GALT, but it's still part of the MALT, still mucosa. It could be any entrance, like females, the vaginal wall has mucosa associated lymphoid tissue. And we have lymphoid tissue all throughout. So we talked about Peyer's patches specifically in the gut, and that's in the ileum. But you can have some MALT and lymphoid tissue in the esophagus and in the stomach and in the other parts of the small intestine. So we have it throughout the whole GI tract. It's just right in the ileum. There's loads of them packed together that those make a distinct area called the Peyer's patches. So the non-specific defenses, you want to be able to identify both external, not all books include it. I just want you to include it as part of the non-specific. External defenses being, don't get in the body in the first place. That'd be like skin, or earwax at that, even acidity of some of the skin of our sweat and stuff. So those would be external things. If you don't get it in the first place, then you don't have to worry about it. So that's one of your best defenses. So then you should know about the internal components. So this slide is probably the best one, giving you kind of a checklist of what's going on in our non-specifics. We talked about the external barriers of not even letting something in. Just the fact that the surface of our skin is dead, that is a huge help because you don't have viruses invading from that, because it's just going to be on dead cells. They can't do anything with that. Phagocytosis, that's obviously the process of one cell consuming another. So we do that in the normal everyday world of fixing repairs, eating up old damaged skin cells perhaps, before we get to repair some skin cell areas. But in the act of that, these phagocytic cells might consume an antigen, and their antigen presenting capabilities would then allow you to alert the specific immune system side. Even though the phagocytic cells are antigen presenting cells, they're in the non-specific side, but that's how you can make notice of the specific side of an antigen being in the body. Inflammation helps bring your immune system components to the site of infection or damage. Natural killer cells, they are a subgroup of lymphocytes, but they are indeed in the non-specific category. These natural killer cells are targeting self cells that have gone bad. So whether they've been invaded by a virus or whether they were malformed and are going to be a cancer cell. So they are ways that we can actually kill some of our own cells for our own protection. Fever is a state that our body gets put in. We reset our hypothalamus. We raise the temperature, our core body temperature, so we make it a lot harder for incoming antigens to proliferate and make more, while our B and T cells are trying to figure out how to fight them in the first place. So fever helps our body buy time, our immune system buy time. Interferon is a chemical that's released by cells that have been invaded by viruses and in other circumstances, but they're ways of our cells to signal out, perhaps the natural killer cells, to identify cells that are indeed damaged and that need to be eliminated and removed. Complement is another chemical process in our body that actually amplifies and enhances any currently ongoing process. So whether it augments inflammation or other chemical activation components, complement will increase that activity. So it's just not a unique thing, it's enhancing what we're already doing, at least, and there's a lot of elements to that, increasing binding and things like that. That's non-specific. So non-specific just means your body's way of fighting an antigen. It doesn't really care who it is. Specific is your B and T cells, but we have B and T cells that are just coded for only a single antigen. So you may have B and T cells that came from a cold that you had in the first grade. So that B and T cell is going to sit around, and you could have measles in your body, and it's like, that is not my job, and it will not do anything to it. So they are specific, and once they code for, identify a specific antigen, that little group of B and T cells will not affect anything else. Anytime you're exposed to an antigen, your body will then make B and T cells that will target and fight that. But the key is the memory cells that come with them. So maybe the cold that you had back in kindergarten or first grade, you still have B and T memory cells for that. And you'll be protected. And if that cold happens to come back around, your fighters have died off after that long of time. So it might take a few days for you to get it up and going, but not the 3 weeks that it would be if you had never been exposed to that before. So it may not be as quick, which is why sometimes when people get vaccinations, they get booster shots because it will help keep the active fighters going, more readily accessible. But even if it's not something that you've been exposed to for a really long time, you will have memory cells for them. So that is the key to the specific immune system defense, is that you create memory cells. So for the B cell family, you will have memory cells, and you need to know about plasma cells. What do plasma cells do? Or plasma B cells? They make antibodies, yeah. So they make the antibodies for whatever antigen that B cell family is targeted to fight, whether it's the cold from kindergarten or whether it was your smallpox vaccine. It's going to make antibodies that will only identify just that specific antigen. On the T cell side, you're going to need to know about helper T cells, suppressor T cells, also known as regulatory T cells. You'll know about memory T cells clearly, and then cytotoxic T cells. Those are the actual fighters. Those are the ones that are like going to bind onto an antigen and snuff it out and remove it from circulation. So the other ones are involved in the process. So you should know about each of those. Immunizations. So active immunization is that you are exposed to an antigen, either through a vaccination, where you get a shot like smallpox or chickenpox or whatever, that's going to be a part of that antigen so that your body can see it and figure out how to fight it and create memory cells for it. Or you just got sick and then your body saw it, created memory cells for it, and figured out how to fight it. So that's active immunity. Passive immunity is when you receive antibodies that another source produced. So it's given to babies via breast milk when they're nursing, or it's given to people that are sick via a serum that came from somebody else's blood that had been exposed to that same antigen before and survived. All right, that's pretty much covers it. So what we went through today are most of the big picture things on those kind of lists. So what they mean is those big areas are, there's usually a grouping of questions that if you understand these big concepts, that you could manage your way through a series of questions. There's a lot of other additional questions. I do my best to try to ask a question from each slide. So when I'm writing test questions, that's what I do. I kind of pop up the slides. I'm like, okay, what do I need to ask from this slide? What do I need to ask from this slide? So there's other details that are out there that we may not have covered today in our big picture ones. The big picture ones kind of gives you the main points that you should have gotten out of each of these chapters, and then make sure you review some of the other elements with just a couple more details.